And now I'm officially welcoming you to EREF Science Session, High Yield and Resilient Anaerobic Co-Digestion of Food Waste and Grease Interceptor Waste, Insights and Applications. And our brilliant speaker today is Dr. Francis de los Reyes III. He's the Glenn E. and Phyllis J. Futrell Distinguished Professor of Civil Construction and Environmental Engineering at North Carolina State University. He's also a university faculty scholar, Associate Faculty of Microbiology, and Training Faculty of Biotechnology, and has been teaching and doing research on wastewater engineering for 25 years. His research and teaching focuses on biological processes and combines modeling, bioreactor experiments, and molecular microbial ecology tools in addressing fundamental and practical issues in environmental biotechnology and environmental engineering. And that's enough from me. Please take it away, Francis.
Okay, thank you. Um, and sorry, let me get out of and make sure that I'm showing you the right thing. Okay. Well, first, I want to acknowledge the students who really did, as the bulk of the work. That's actually what happens in research labs at universities. Dr. Ling Wang is a former PhD student, and she's now with the FDA. Um, um, Emma Gwertin was a former master's student, and she's now working with Brown and Caldwell. And then also Giovanna Portioli was also a master's student, and she's now with the city of Charlotte. So we'll talk about food waste, and we'll talk about the potential for food waste for anaerobic digestion and some of the insights that we've learned. So we basically waste billions of pounds of food in the U.S. One estimate is 92 billion pounds of food waste. This was a number from Feeding America. The EPA a few years ago estimated this as 66 million tons. And so the number kind of varies a little bit, but that's about the right range. And you probably heard the statistic that about 40% of food from farm to fork goes uneaten. So it's really a lot of waste. That's something that has to be done about. One particular waste is grease interceptor waste. And this is the waste that goes into grease interceptors from food service establishments. And a few years ago, we estimated that to be 5.8 billion gallons every year. And that's a lot of energy. And that can actually be used if we can use all of that embedded energy in grease interceptor waste. That's a lot that we can use to maybe even power the water and wastewater industry. So the EPA put out this uh, mass balance. It's a flow of wasted food generation and management. And as you can see, the um, the food coming from retail, from food service establishments, so these are restaurants, and then households, a lot of that actually goes to wastewater treatment. About 59.8% goes to landfills. And then I highlight there at the top, 0.81% actually just goes to anaerobic digestion. This is data from 2019. So there's still a lot that goes into landfills. And so I hope that we all agree that there is potential for anaerobic digestion, and you can make a case that it actually makes a lot of sense. So, for example, if you do a life cycle assessment, this was data from work here at NC State from 2011 with Mort Barlas and Jim Levis. They showed that anaerobic digestion is the most sustainable waste management approach for food waste compared to, for example, composting. We know that many US states, at least eight now, and there probably will be more, who will legislate the prohibition of landfilling of food waste. Um, I just saw actually a news item just this week that there was a paper that evaluated the status of this for 5 states, and they found that only Massachusetts actually was doing a good job of actually diverting food waste from landfills. And they said it had to do with more stringent regulations and also the availability of composting facilities. So even though they're diverting it from landfills, a lot of that food waste still goes to compost. If you look at food waste, the data from EPA is basically saying that most 58% of methane emissions uh, from municipal solid waste landfills is from food waste. And if you think about the potential, a lot of utilities, so we're talking about water resource recovery facilities or wastewater treatment plants with anaerobic digesters, they have underutilized capacities, and but they're not, capturing the methane because of low yields. So first, they may not be accepting food waste or grease interceptor waste, but then also they're not capturing the methane, maybe just burning it because of low yields. So the idea is that if you divert your food waste to these digesters, you actually can increase the yields, and then it will make sense to capture that methane for energy. Just this year, the American Biogas Council estimated that there are about 110 standalone food waste AD facilities, 45 of which are post-consumer, and then the others are pre-consumer. So, they counted places like breweries, for example, because breweries, as we know, um, the standard technology is actually anaerobic treatment. This is a table from the US EPA. They're still conducting the survey for this year to reflect 2023 numbers. So this is reflecting 2020 and 2021. Um, so they send out these surveys to their list. And what's come back are these estimates. So you can take a look at the first column, digester type. So the first is standalone digesters. So these are actually built to take in food waste. And then on-farm digesters, and then the water resource recovery facility co-digesters. So these are the existing anaerobic digesters for biosolids treatment, but actually they take in food waste. So about 15.8 million tons in 2020. Um, and then the third column, they take out the beverage waste. And what you can see is that the total, uh, is basically something like 16.9 million in 2021. Um, but the bulk of it is actually in terms of beverage waste. The American Biogas Council again has estimated that there are about more than 1,000 facilities with anaerobic digesters, but only about a third, 360, are currently co-digesting food waste, and mostly these are grease interceptor wastes. So there is a lot of potential there. Two-thirds can potentially accept food waste. The picture here is the Maryland Bioenergy Center that actually takes in food waste, and they're taking in around 100,000 tons annually. Okay, so the motivation for the research that we've been doing, and I'm actually going to summarize several research projects, just trying to get to the results as quickly as possible so we can have a lot of time for discussion. But the motivation is to increase economic feasibility. So a couple of things there. One is we need to maximize methane yield. And by that, we mean the amount of methane we get for every maybe gram of volatile solids or gram of COD we put in. Because that methane yield directly relates to energy production and, making our economics, turn to the right, to the right hand column, which is the actual profit, right? And number two is this question of food waste is really not a monolith, right? There are different kinds of food waste. But what happens when you change your substrates, both in terms of food waste types? So you go from, let's say, more proteinaceous waste to more food and vegetable waste, which is more carbohydrates, and you're going back and forth. And then also you're changing loading conditions. So the scenario here is that, you don't really have a choice as to what kind of food waste comes in every single day or every single week. Perhaps, the local grocery has a couple of trucks of waste cabbage or lettuce, or the next day a bunch of rotten, meat. Um, how do you maintain a high and stable methane yield if you're basically perturbing or putting this stress into your anaerobic co-digester? So these are the questions that we have, right? And then there are two scenarios which correspond to kind of like the types of anaerobic digesters that I showed earlier. One is the scenario at the treatment plant where you're co-digesting your food waste with biosolids, in this case, thickened waste activated sludge. Or scenario two is if it's a standalone digestion or co-digested with municipal solid waste. So you've got these two different scenarios, and do you get different performance depending on which kind of reactor you're using. So let's talk about grease interceptor waste. This is a grease interceptor at the parking lot of a food service establishment or restaurant. Typically, the top layer is the fog layer, fats, oils, and grease. And then food solids settle to the bottom, especially if there's like a garbage disposal unit. And then the water is somewhere in between. So that's what happens in the grease interceptor. You actually want the fog to rise to the top so that it doesn't go into your sewer line. And then maybe, once every month, once every 2 months, the whole thing is emptied, and that's your grease interceptor waste. So there's a fog layer, and in the lab, you can separate these layers so you know exactly the proportions that you're adding them in, right? Because the food particles are very different from the fog layer. But there is a problem when you're co-digesting this grease interceptor waste. Number one, it's high in fats, oils, and grease, so you can think of that as lipids. And if you have overloading of this, you basically sort of push the system one way. And as you know, in anaerobic digestion, you want it to be a very balanced network, where you have your hydrolyzers, your acid formers, your acetate formers, and then your methanogens. And as you know, having this right balance of microorganisms is key to having a stable anaerobic system. But if you overload with grease interceptor waste, you are basically pushing it one way, and that stress can cascade down the system. You might have a rapid increase in long chain fatty acids and volatile fatty acids. And we also know that there's long chain fatty acid toxicity to methanogens and other organisms. So, for example, there's damage to the cell membrane, and then also loss of methanogenic activity. So that is an issue, and we want to make sure that the right organisms, bacteria and archaea, are in your system. So that's the challenge. What I'm showing here is a summary, if you will, of where we are in terms of yield, methane yield. So the y-axis is methane yield, liters of methane per gram of volatile solids added. And we can sit here and argue that volatile solids is really not a good denominator for this, right? Because you can imagine the COD per volatile solids of lipids will be very different from that of carbohydrates or for proteins. And I agree. I think the denominator should be gram COD. But as it is, majority of the previous work has been in terms of per gram volatile solids. So that's the y-axis. The x-axis is the organic loading rate, basically how much we are putting in, and this is fog-based waste. So we're looking at all the literature that only are on the fats, oils, and grease in terms of grams per liter per day. So loading rate of the reactor. And these are some of the different studies that have been done. The first thing I'd like to point out is that there seems to be an increase up to 1 gram VS of fog-based waste per liter per day. I'm going to call that zone 1. As you increase the organic loading rate, you get an increase in the methane yield, and that makes sense. And then in zone 2, um, greater than 1 gram per liter per day, you can get an increase to maybe around 1.5. But notice that there are also instances where you get down to zero. In other words, in these studies, they actually killed off methanogenesis. Um, so anything above one, you might either get an increase or you might kill your reactor. And as you can see, many examples. So the question is this the highest we can go? 0.5, zero point 0.6, and what makes a reactor actually be able to go higher? So that's the question we're going to try to answer. So there's a bunch of other work that we've done, but I'm just going to show you one set of experiments first, where our hypothesis was that we could actually make the microbial community adapt to grease interceptor waste using two approaches. One is step feeding. And with step feeding, what we mean is we keep increasing the organic loading rate in stepwise fashion, kind of like a ladder. Now in the graph here, you can see it actually is kind of like going up and down, just to reflect that we actually feed it every 2 days in these examples. So there is if you will, a spike every 2 days. But the bottom line is that it actually is a step load. On the right is another type of adaptation. It's pulse feeding. So you have a base load. And so again, you have this every 2 day spikes, but then you give it a higher spike, a big pulse, and maybe that's another way of adapting the community. So the hypothesis is that you're getting the right microorganisms, maybe the methanogens, maybe the syntrophic bacteria and other populations to actually be trained and increase in the system. And by the way, this particular project already has been published, and I neglected to put the paper there, but it was in water research by Ling Wang. So just to give you an idea of this particular experiment, this one is simulating, again, an anaerobic digester at the wastewater treatment plant. You have a control, you have a treated, and you have a way of capturing the gas and measuring how much methane you have using, a GC. And you can also measure things like VFAs, obviously pH, and so on. I just wanted to give you an idea. That's why I'm showing this picture. Okay, let's go to the results. Um, let's focus on the top row first, and that's the organic loading rate. So the control digester is the black line, and it's basically just waste activated sludge, thickened waste activated sludge. So there's 0% GIW added, and that percentage is on a weight basis. Now look at the gray. So after about 90 days, you start feeding the grease interceptor waste to the treated digester. First, you add 46% by weight. After a few more days, you add another step, 66%, and then you add another step to 84%. Notice that from 84%, we went down to 0%, and that's because of the methane yield. So take a look now at the second row, the second graph there. And the gray again is the treated digester, and this is the methane, and it's normalized per gram VS added. The control again are the dark lines. You can see it's pretty stable all across the whole experimental period of 200 plus days. And then as you increase the loading, 46%, you get an increase in the methane. 66%, you get an increase in the methane. But then when you actually went to 84%, the methane yield went down. And so the decision was made to stop feeding it, let it recover, get to 0%, which is what we have here. And then there's additional things that we did in phase 2 and phase 3. Basically, um, the the idea here was that the control reactor was also hit with a higher load, and it decreased in methane. But then when you let it rest with 0%, you get an increase. In other words, it was able to process the grease interceptor waste that was fed before. And then when you hit it again now with 66%, you were able to increase the methane yield for the erstwhile control reactor. In the meantime, the other reactor, the gray one, went from 0% to 66%, and it stabilized. And when you hit it with 75%, it also increased. So In other words, there was some training going on. And again, the biogas production and the methane content, these were all consistent with the results. So what does this mean? And again, I'm summarizing a lot of data here, jumping to the main conclusions. We measured VFAs, we measured pH, we measured all of these things. Um, but let me just show you what this means. So going back to our GIW co-digestion map that we had before, um, you can see that if I start with step feeding, this is the black triangle. If I step feed, I can get to a higher methane yield. Another step feed, a really high methane yield, right? Above zero point 0.7. This is the black triangle at the very top. But then if I increase it some more to 84%, it actually went down a little bit. So step feeding, I was able to train it, but there's still a maximum, if you will. For pulse feeding, and then actually it goes, again, that's another step feeding arrow there. For pulse feeding, you can start with, um, the 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 square here, the open squares, and again you can pulse it, and you can see it increase and increase. But if you increase too much, it actually goes down. So what this is showing is that there seems to be a way of actually getting to very high methane yields, but you've got to train your reactor. And there is a way where you can do the step feeding properly, but if you step too much, it's going to go over the over the top, right? It's going to crash. So based on this data, we think that about zero point 0.6 to zero point 0.7 organic loading rate per jump is sustainable. Anything higher than that is going to be it's going to be too fast. So you can see that when the jump was too big, it failed right away. But if the jump was small enough, you can actually train your reactor. So that's the thing is and I think to date this is probably still the highest methane yield in recent step to waste co-digestion. So the second question is managing the different types of loads, meaning um, if you have, again, different types of food waste, protein or carbohydrate or lipids, what's the highest loading rate for each of those types? Are there specific communities associated with those substrates, and how do these communities shift over time in batch conditions? And then when I do change, let's say from protein to lipids or lipids to carbohydrates, what happens to those communities? And then additional questions are how do we ensure replicability, and then solids versus leachate communities. I'm not going to touch on those questions today, except to say that we have a system where we basically have replicates, and we basically have destructive sampling to make sure that we can sample the whole reactor but still have replication. And at the end of the day, this is really the question, right? How do we influence these communities by adjusting feed types and avoiding, again, these detrimental shifts and combinations? So the first one is the highest methane yield for different food waste types, and the second one are the shifts in methane yield. Um, the idea is summarized in this um, ternary diagram, this triangle. And each of these points of the triangle are the different kinds of waste. So 100% cellulosic fruits and vegetable waste will be at the top vertex. Lipid waste will be on the left, and protein will be on the right. But then you have these various combinations, right? So fog plus fruit and vegetable waste, and so on. So what can we say about all of these different areas? That was the dream, right? Is to actually look at this picture and be able to say something about each of these areas. So I'm just going to show you very quickly. The student had to, you know, chop up the meat waste, cook them, and also the fruits and vegetables, and also collect receptor waste. These were based on USDA, US consumption data. We did characterize the feed in terms of solids, VS to TS ratios, and COD. And so we're shifting now from a VS loading to a COD loading, even though it's quite difficult to do that for lipids, right? Um, So the first experiment is a wet system experiment, very similar to what I showed you earlier. This is co-digestion with thickened waste activated sludge. And the second experiment is a dry system experiment. This time it's co-digestion with MSW. Um, I'll first explain the wet system. This is very similar to what we did before, uh, where you have protein, carbohydrate, and lipids, and you basically do step feeding, or actually it's more of a, yeah, step feeding, um, and increasing the load and trying to see how high can we go. That's basically the question for protein, carbohydrates, and lipids. And I'll show you the protein and carbohydrate data. Just to show you what it looks like, these are the systems that we did in the lab. It's all in this hot room at mesophilic conditions. We used anaerobic sludge from a wastewater treatment plant, and these are duplicates, about zero 0.6 liters, fed every 4 days, and retention time, solid retention time of 20 days. So let's go to the results. Again, you can see the very top row, you have the loading rate. You basically, again, have this step feed happening. And then the methane is the second row. And you can see that it's increasing stage 2, stage 3. When you get to stage 4, you get this erratic behavior in terms of the methane production. And you get to stage 5, you actually kind of see a leveling off of the methane. So it's not about, it's not commensurate with the increase in the load. We did achieve the organic loading rate of about 4 grams COD per liter per day, and the highest methane yield is about 169 milliliters methane per gram COD. And this is a little bit different than per gram VS. But it was an 80% increase compared to just the control, which was the waste activated sludge. The other thing we did is we sampled this as this was going on and used DNA sequencing. The bottom line is we can tell based on the sequencing of the 16S rRNA gene which organisms are there. And you can see just from looking at the pictures. So each of these bars is actually a snapshot, and each of the colors corresponds to, in this case, a genus. And you can see that the composition changed over time, right? You can see the colors change over time and the community is shifting. When we plot this on top of the methane yield data over time, you can see when we started out, for example, you've got the methanosida there and this organism called Tetrasphaera. And the first, a few days until stage 2, that's more or less those are the dominant organisms. But then you get this pink Peptostreptococcus, which is actually a meat. It's part of the Carnobacter. It's actually a meat uh, eating, if you will, organism. And then also Actinobacteria, which is this green color there. So there is this shift as you keep, uh, moving along. What about the carbohydrates? So here the carbohydrates, a little bit different story. From stage 1 to stage 2, you have an increase to stage 3, but then in stage 3, the methane dropped, and so you have a failing reactor. So we return that to no loading, trying to make the reactor recover, and then pushed on with stage 5, 6, 7. And as you can see, what's interesting is one reactor actually responded well to stage 6, but one other reactor kept failing, kept decreasing in terms of methane. And that tells you that maybe at this loading, um, even though those two reactors were operated the same, one went one way, the other one went the other way, and you're very, very close to the limit, right? So a little difference in those two reactors led to one failing and one succeeding. Again, the numbers are here. We did get a higher methane yield, about 149% increase compared to the control. But there was a failure in stage 3. It did recover. Um, and then when it recovered, you were able to push it again with higher loading. If you look at the microbial communities, it's very clear that you get a switch right away with the fruit and vegetable waste in terms of the microbial communities. So these organisms, The tan one is Trichococcus, and then it shifted to the dark brown one towards the end there of stage 5 to Paraclostridium. So we're still analyzing this data, but these organisms are organisms that can handle sugars and carbohydrates. But it's really interesting how the shifts correspond to the functional changes happening in the reactor. And there's a lot more we can say about that, but let me move on to the conclusions. Okay. So in these high protein reactors, we can actually get to very high ammonia levels, and the carbohydrates, even though they failed, they can actually go back. And I didn't show you the lipid reactor data, but that really achieved the highest methane yield. Okay, so now let's move to the dry system. This is a very unique experimental design. On the left, you started with all protein, but then some reactors were switched to carbohydrates. Another reactor was switched to lipids, right? And the rest, we increased the protein load. And then those increased protein load reactors, some were shifted to carbohydrates, others were shifted to lipids. So now we're actually shifting the substrate. And along the way, we're destructively sampling so that we can get the DNA data. So that's actually how this experiment was set up. So again, just going back to the results, start with the brown. You can see the cumulative methane increase over time. Once it reached um, or it was starting to plateau, we basically sacrificed some reactors, opened them up, and then switched to the carbohydrates, which is the green, or to lipids, which is the yellow. And so for some of these reactors, they were destructively sampled, so the plot stopped, and the going back to zero is really just restarting by feeding it and then measuring the gas again. Um, and you can see the differences between how fast the carbohydrate responded, but how high the lipids actually increased. So, okay, so some conclusions. Um, when you switch from protein to lipids, you get the highest methane yield. But when you switch from protein to carbohydrates, you actually have a faster increase. So it took only 11 days for the carbohydrates, that was shifted to reach 50% of the theoretical methane yield. Another finding was that the reactors, when you kept increasing the protein, were able to withstand ammonia concentrations up to 10,000 milligrams per liter, but at around 15,000, you get inhibition. So um, again, this is a function of pH, but these numbers are really interesting as to how high you can load with the protein. The next experiment was again trying to imagine this ternary plot and trying to figure out what's the best combination. And so this experiment was unique in that we started with the vertices, 100% carbohydrate, 100% fat, 100% protein. And then for some reactors, you move them to points D or point E or point F. So basically, you're trying to cover all the space starting from the vertices, eventually ending at G, which is in the middle. So it's a little bit complicated. There's a lot of reactors involved. There's destructive sampling involved so that we can get the DNA. But the idea was to cover all of those spaces and look at the methane production, and then also look at the microbial communities. Okay, so just to give you an idea of what that looks like and how many reactors were involved, this is one run. Again, in this room that's at constant 35 to 37 degrees centigrade. I just wanted to show you some data, what those reactors look like uh, in terms of the typical batch performance, or where the methane goes up and then goes down. And then when you switch, so around day 39, um, the carbohydrate ones were then switched um, to carbohydrate. Sorry, the other vertices were switched to carbohydrates, and you see the increase. And then where the blue line is where the fog and protein switches happen. And so again, you get this increase and decrease. And it's a lot of lines, and there's a lot of analysis. But one way to summarize all of that is this ternary plot, where you can actually look at the cumulative methane. So here, the darker the red color is, the higher the methane. And this is what I was trying to envision when we looked at ternary plot and we said, can we plot where the regions of highest methane are? So the highest methane yields, it turns out, are in between fog and carbohydrates, and also in between fog and foods and vegetable waste. And interestingly, if you have a balanced diet, if you will, in the middle of this triangle, you actually have the lowest methane yields. Um, so we were able to cover the space and actually apply a mathematical model so we can actually take a look at these areas with the highest methane. And then, and again, this is going to take a lot of time to go through, but I'll just try to show you the big picture. If you plot the microbial community structures in this triangle, you can actually see the differences. So, for example, we said that point F right there at the bottom between fog and protein, that's where some of the highest methane yields are. And that's the microbial community, um, uh, mostly Methanothrix, Methanoceraceae, and some Clostridium. But if you look at point G, look at the microbial community there above G. It's very diverse. There's a lot of organisms in there. But remember, those are some of the lowest methane yields. So it turns out, and again, if you look at between fat and carbohydrate, point D, that community looks very similar to point F, right? And again, that's some of the highest methane yields. So we can actually take a look at this and correlate the microbial communities with the methane yield and what's happening in this space. And there's a lot there. There's a lot that we can talk about. Again, in the interest of time, I'm jumping to the main conclusions. So the highest loadings to initial failure, depending on your type of food, um, are given here. Uh, and if they initially fail, they can recover. The highest methane yield was for lipids, which, as you can imagine, has a lot more energy. We can add protein addition to around 29 grams COD per liter. After that, you get ammonia inhibition. The worst resilience, meaning you'll get failure, is when you start with proteins. But the best substrate shift is from proteins to lipids in terms of getting the most methane out. And then the best combinations for maximum methane, like we said, was lipids and proteins and lipids and carbohydrates. And in fact, the balanced diet in the middle is not actually the highest methane yield. So some implications for operation. One is don't start with proteins and avoid proteins alone. So if you're starting out your co-digester, don't start with putting a lot of meat waste in it. Um, it's much more favorable to add fruits and vegetable waste or grease interceptor waste, FOG, to proteins. And it's okay to add proteins to fruits and vegetables or FOG, but not proteins alone. And it's also good to add FOG to proteins or fruits and vegetables. Um, and again, the balanced diet, if you will, doesn't yield the highest methane. So this is my last slide, and just to summarize everything, with a series of experiments over several years and funding from EREF, we were able to figure out, some steps to get to the highest methane yield if you're only doing grease intercept to waste. In other words, if you can train your reactor with either step or pulse feeding, you can get the highest methane yields. But you've got to mind your step. Too big a step is not very good. Um, and obviously, if you acclimate it to grease interceptor waste, it leads to more resilient anaerobic digestion of grease interceptor waste. What you start with is important. And also, when you're shifting your substrate, it's also important. So let's say you currently have a lot more proteins in there, you shift it to lipids, you're going to get a real good increase in methane. And then finally, if you have a chance to combine your wastes, then you identify some of the areas that will get you the maximum methane. Um, Maybe you don't have a lot of choice, but if you have a way to store your truckload of meat waste for a little bit, you can play around with the combinations so that you can maximize your methane yield. And with that, I hope I left enough time for questions. And thank you very much for your attention.
Thank you, Francis. You really have us thinking and shared a lot of great data and research. And before I get to questions, because we have quite a few, I just want to let everyone know that we'll be sending a link to his presentation in a follow-up email. There's a little glitch when we were loading it, so please look out for that. And then, Francis, I'll get into the questions.
Okay.
Um, the first one is, what do you consider the absolute minimum facilitating conditions for this type of project to be economically successful?
Oh, that's a good question. We actually have a paper um, that has done a techno-economic analysis. We're not quite finished with the paper. We've been working on it for a few years now. Um, but in that paper, we did a techno-economic analysis, and based on what you're starting with. So, for example, if you're a large wastewater treatment plant with an existing AD, where are your cutoff points in terms of costs and benefits, the break even. If you're a treatment plant and you want to build a new um, co-digester, what will be the cutoff points? And so the answer is really it depends. And I'm sorry, that's an answer that usually you hear, right? But it really depends on what you're starting with, what your current capacities are, and there's not a single one answer because the break even curves look different depending on those different conditions. So context does matter, and I hope that we can get that paper out soon because it's a very useful way of looking at again costs. Yeah.
That makes sense. Is pre-processing of the mixed food waste required before going into the digester?
So that's a good question. For our experiments, there was some pre-processing in that we chopped up, because these are small digesters in the
Hmm.
lab. So we did chop up the meat. You saw the pictures. We did kind of chop up the vegetables. Um, in any of these systems, if you have a way of making the particle sizes smaller, that's always better for your systems. So for more efficient methane production, yes, some way of pre-processing in terms of chopping up to just smaller pieces is needed. But you don't really need any enzymatic treatments or anything else. It's just making the particle sizes smaller.
Okay, thank you. And can you also discuss upper organic loading rates observed in full-scale
Yes,
co-anaerobic digestion? Okay, thanks.
sorry. So typically the upper organic loading rates, like I showed in that graph, are in the, one, maybe slightly higher than 1 gram COD per liter per day, or gram VS per liter per day. And again, the one of the issues with this in our field is that we have traditionally used volatile solids, and that's really not a good way of. It's an easier way, but the problem is the gram COD per volatile solids of lipids is very, very different. That ratio is different from that of, let's say, fruits and vegetables. And so, you know, we are opening ourselves up to um, errors, if you will. But roughly speaking, if you're in the one range, gram vs per liter per day, you're probably going to be in a good operating condition. If you want to go higher than that, then that's where some of our recommendations would really make sense. Meaning, you can do a stepwise increase, mind the step, the size of the step, and then also make sure you are using what I call the right mix of substrates, so a little bit more lipids than just proteins.
Okay, thank you. When is fugitive methane a concern from various AD systems?
It's 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 always a concern.
Mhm.
What's happening right now in digesters where um, I mean, we know this is that if they don't have a way of capturing the methane, which requires gas capture, gas cleanup, and so on. Um, if the economics of that is such that it's more expensive to put in those facilities, then at treatment plants they just burn it, right? They just flare the gas, at least convert that CH4 to CO2. And that's why the argument is if you have these standalone reactors, obviously you're trying to capture the methane, or if you have co-digestion, you're increasing the yield so that it makes more economic sense for you to go ahead and buy the equipment so that you can reuse the gas. Um, that's where this techno-economic analysis will be more important. Fugitive emissions, um, is something we should avoid in general in the industry. I know there is a study that just came out in terms of emissions from landfills and ways of quantifying those emissions. And so that's a very important field. But it's always a concern, and we always want to make sure that we think about that. One of the issues with putting food waste into landfills is that, and the reason why food waste accounts for a lot of the methane coming from landfills is that, with food waste into landfills, you get the methane conversion quickly, and maybe you don't have the gas collection set up yet in your landfills. In other words, you get the decomposition happening faster, um, and then you've got these fugitive emissions, if that makes sense. Now, if you have a standalone reactor, you design it from the outset where you collect the gas. In a landfill, you know, there's a lag between when you are operating the landfill, filling it up, and then making sure you have the gas capture.
Thank you. And then this one's an interesting one, Francis. Um, is there a way to prevent microplastics and food waste slurry from entering wastewater treatment plants? Has there been any studies on the fate of these particles yet?
That's a really good question. And I wish I were more up to date on that literature. I I'm gonna confess, I'm not up to date with the literature. I know it's an issue,
Mhm.
microplastics, and the other thing that you might hear, obviously people will hear about is PFAS. Um, so yeah, I mean, I think there's a concern there. You know, uh, in the landfill, the question is again partitioning to the solids phase. Does it
Mhm.
go to the leachate? The leachate is treated. Um, what's the fate of that in your treatment system? So these are all valid questions, and unfortunately, I'm not able to give you an up-to-date answer on that.
Oh, that's okay. It's something that will be researched in the future, so I'm sure we'll come back to you.
Yeah.
And then, how do we ensure environmental sustainability in this climate changing state?
I I think that actually diversion, the reason why a lot of states are diverting food waste from landfills is because they understand that uh, this is contributing to methane emissions. As you know, methane is a very powerful greenhouse gas.
Mhm.
So if we are actually able to use anaerobic digestion as a way to capture that methane, reuse that energy, then we are already preventing, methane emissions, and then we're also getting a benefit from the energy that we can get out of it. So I do think, just like that LCA paper from
Mhm.
2011, I do think that anaerobic digestion is one of the most um, uh, beneficial, most efficient, effective ways of you know of managing food waste. So I think the issue is always, does it make sense based on the context? Um, in other words, do I have the capacity for that in my existing plant? Does it make sense to have a standalone system? And I think as more and more states put out new regulations and make it more stringent, then there's more and more incentive to actually put on this, standalone digesters. As you saw that one in Maryland. We have a few of them around the country, um, and that's really, the best way to make sure things are, sustainable. The other part of this also is just minimizing food waste. That number, 38% of wasted food, and some of it is pre-consumer. A lot of it is also post-consumer. I think we need to think about that also, because that's just inefficiencies, right? If you think about it, that's a lot of food that we waste. It doesn't, get uh, to to feed people.
Right. So true. And then if we're balancing co-digestion with nitrogen discharge limits, do you have a sense of which feedstock combination best balances methane yield with nitrogen yield after dewatering?
Oh, that is such a good question, and I didn't touch on that. Obviously, in this case, proteins again are bad because proteins actually increase the ammonia um, that is going to come out, right? So if you think about the whole cycle with your anaerobic co-digestion, you're still going to have the digestate coming out of that, and then if you actually dewater, you're going to get that liquid. Um, that side stream is going to be high in ammonia, just because it's a digester, right? That's what a digester does. Your biomass alone is going to contribute to ammonia. But then if you had food waste like high protein, meat waste, you're going to get an increase in that ammonia. And like I said, in the reactors, 10,000 milligrams per liter. Um, so that's going to be an issue. In some treatment plants, like in the city of Raleigh, a side stream system is something that they're already thinking of. They have a different process. They have thermal hydrolysis of the biosolids, so they expect a lot more increase in the nitrogen in that liquid. So that's something that we have to think about in the context of the wastewater treatment plant. The flip side of this is, I, I don't want to say protein is bad. Protein is actually good when you think about the carbon to nitrogen ratio. Just making sure that that ratio is in the optimal range. So something that I did not report on in my talk, but that's something that also needs to be measured and um, making sure that the reactor is operating well. But again, that side stream is going to be an issue, um, and different treatment plants are coming up with different ways of treating that. For example, using Anamox, or partial, night denight processes.
Okay. And here's another technical one. Have you tried maintaining the substrate mixture that obtained max methane yield for a prolonged period? And does that yield remain constant or does it eventually drop?
Um, we have not, so prolonged period. Um, so in these experiments, you probably saw the x-axis. It was in the order of 30, 40 days. Um, but if you're talking about running something for 200 days. We have not done that. Just trying to maintain a reactor at that optimal period. So yeah, the answer is we have not done that in the lab. I don't have any reason to think that it's not going to be sustained. Um, I do know that in practice that may be a little bit hard, just because again, one day you're getting a truckload of pickles, another day you're getting a truckload of, I don't know, olive oil, right? So
Hmm.
it's just you're not in control of that, and if you are accepting everything, um, then maybe, and then you don't have a lot of storage, then maybe you are kind of at the mercy of what's coming in every day. So that's where the dynamic changes. That's where kind of like we're interested in, if I understand a little bit about what I can do and what I shouldn't do, that's going to guide me at least in terms of either accepting certain loads or having a way to mix different kinds of food to make sure I don't hurt my reactor.
And that's what he was asking. He said it appeared some microorganisms respond better to different waste streams. And he was
Right.
wondering if you could introduce the better performing organisms with
Oh,
corresponding waste to keep yields from reversing.
oh. So this is bioaugmentation, right? Um, um, I have mixed feelings about bioaugmentation, to be honest. We actually had a paper in a different context showing that it didn't work very well. Um, I think a lot of the organisms you need are already there. It's just a matter of making sure that they're happy, and maybe that once in a while you might have to shock them also. Not shock, but perturb them, to make sure that the right organisms are in your system. Um, uh, so yeah, I think bioaugmentation works under certain conditions, and then in some conditions it doesn't work. Um, I guess that's yeah. So to me, it's not 100% foolproof to add organisms. I think it's better to think about the organisms I have already there. How do I actually keep them happy?
Okay. And then where would the food waste enter the process in a treatment plant with influent or added in into the reactor?
Yeah. So I think the easiest way is actually added to the reactor. Um, yeah. So you would have to think about the how it's added in, the design of that structure. You might have another uh, container or tank. You might have pre-processing, like we talked about, maybe some grinding, a little bit of that before you feed it in the digester directly. That's where I would imagine it to be the simplest, and maybe the best. Otherwise, if it's in the influent, then you have to think about second order effects on your wastewater treatment plant itself.
Okay. And then I think we have time for one more. Why is composting the preferred method for food waste for many munis over AD?
That's a good question. um, I think I'm going to give you a general answer first. The general answer is that uh, AD has gotten a bad rap because um, of some of the failures, right? I think most people say, "Oh, it smells," and or yeah, I mean the smell, or it doesn't work, and a lot of that has to do again with not understanding that it is a process, and that process has to be maintained and operated. So, for example, if you do get fatty acids like butyrate accumulating because you overloaded it, or you didn't watch the pH, or you didn't handle it right, then yes, you're going to get smelly, digesters. So I think that has gotten a bad rap. But my response to that is it's a process, and you have to manage it. It's a biological process, so you have to understand what's going on. And if you do it right, it's very stable, it's very economical. Um, it's kind of like any other machine. If you don't expect your car to run without doing regular oil changes or changing your tires, right? So you got to have this maintenance, and you got to have the proper controls. Um, so that's the general answer, right? And we have to work to actually make sure that that's not what most people think about, is that, oh, anaerobic digesters compared to composting, where you know we it's aerobic. I turn it over, then it's going to work. um, yeah, and what we're seeing is that, again, in some states, the lack of composting facilities, even though they do have the regulations, it's not as effective in terms of food waste diversion from landfills.
All right, thank you so much, Francis. You gave us so many more insights in the questions as well. So thank you, everyone. This concludes EREF Science session for today. And once again, big thanks to Francis for his time and expertise, and of course to all of you for joining us today.